Thank you very much for the opportunity to present Graybug Vision today. Our technology platform is based on a very simple injection technique which does not require any surgical intervention. It's a small gauge needle that is used to deliver our formulations. And it's a highly tunable platform. We can tune the rate of elution of drug as well as the duration of action. We've tested our technology in multiple routes of administration, mainly IVT, but also subconj, and we have a very strong family of patents around the technology. I'm not going to go into the details of why we want to address these challenges of repeated administration of drugs for diseases like retinal disease, but also glaucoma. I think my colleagues have very well explained this morning why there is such a large unmet medical need, despite the presence of very good treatments currently. We have currently 3 programs that we are progressing. The first one is called GB102. It's a twice-a-year pan-VEGF inhibitor formulation that has gone through a phase 1/2A study, which I'm going to talk about later. We have a similar molecule, same molecule with a different formulation, GB103, which has a 1-year duration, as well as a proprietary molecule in glaucoma, GB401, which we are designing to last between 4 to 6 months. We are working on building an additional pipeline of drugs. The technology was originally developed at Johns Hopkins. We still have a research lab in Baltimore, and you have here the syndicate of investors supporting us. We're a private company. So GB102 is a depot of PEGylated PLGA microparticles containing Sunitinib, and we like Sunitinib because of this pan-VEGF action that it has, blocking all VEGF receptors, not only VEGF-A, as short-acting anti-VEGF currently do. There are 3 levels of innovation we delivered at Graybug. The first one was to make these PLGA microparticles non-inflammatory. As you know, PLGA is a very good polymer, actually releases drug in a very controlled manner, but historically created some inflammation issues. So we have a technology to make these microparticles not inflammatory. The second piece of innovation was to ensure these microparticles were not going to float in the vitreous and create a snow globe effect when injected, but were finding each other, aggregating, and creating a nicely formed depot in the eye. And the third piece, of course, was to use successfully Sunitinib in CNV. So this is how it works. You have a suspension of microparticles being injected in the vitreous. When the drug gets in contact with the vitreous at 37 degrees, it starts aggregating, forms a depot, and from the depot, the drug elutes over time. Depending on the formulation for GB102, the drug will elute for a number of months, and then the polymer is being hydrolyzed and just washes away from the vitreous. There is nothing left behind. So we spent a lot of time optimizing our formulation and our presentation. So now this is how our drug presents. So we have a vial containing a lyophilized microparticles and a prefilled syringe with a diluent. You just inject the diluent in the vial. It's vortexed, and you extract the quantity to be injected. And this is a video, maybe not. It's supposed to be, yeah, here it is. So this is a video of the injection. So it's a normal IVT injection with the exception of the speed of injection. So we inject over 5 seconds to ensure that these microparticles aggregate straight out of the needle and form this depot immediately. And here is at day 1 in the rabbit eye. You see the depot very nicely formed. And by day 98, if I can go to the next slide, but at day 98 in that particular model, you could see the polymer was entirely gone. I'm sorry, I'm trying to move to the next slide. Here it is. So we conducted a phase 1/2A study, which was done on 32 patients, 4 different doses, single injection on patients that were previously treated with anti-VEGF, normal short-acting anti-VEGF, Avastin, Lucentis, or Eylea. And you can see after a single injection of GB102, the vision gains obtained with the short-term short-acting anti-VEGF were maintained over 8 months, and so was the anatomy of the retina with a nice CST maintained over 8 months. and you have the patients that were only injected with GB102 without rescue in orange. We looked at the rescue-free numbers, and basically what you have here is at 3 months, 88% of patients did not need any rescue with short-acting anti-VEGF, 68% at 6 months without rescue, and even 42% at 8 months, which is above our target product profile. The rescue-free rates range from 50% to 88% of patients without rescue at 6 months, depending on the dosages, and no dose-limiting toxicity. We are moving now into 2 phase 2 studies this year, a phase 2A in macular edema, starting very soon, and a phase 2B in wet AMD with a control arm using Eylea as indicated. And lastly, we have also, we are very excited by this glaucoma program, and if you focus on the left-hand side only, we've conducted this animal study showing that after 1 injection of GB401, we could lower IOP at levels equal or actually below what you normally get with the topical use of an eye drop, and we are planning to move this program into the clinic next year. This is how our pipeline looks like, and I think I have used my time.
You can summarize.
Thank you. As a summary, you have, we are moving into phase 2 this year. Thank you.